Friends, welcome back to your channel Bioexam and Research Support by Archana. So in the last video I have told you about the C-value paradox, that is the lack of correlation between genome size and organismal complexity, and I also discussed that it is because of the presence of repetitive DNA sequences. So in this video I'm going to discuss about the repetitive DNA sequences and what are repetitive DNA sequences and what are their functions, why they are present in DNA, are they junk DNA or not, and then I will also discuss about their types and examples. So if you are new to the channel, take 2 seconds and just subscribe the channel so that you won't miss any update. Now let's get started. So repetitive DNA comprises DNA sequences that are present in multiple copies throughout the genome, and these sequences are largely non-coding, means they don't code for protein, and yet they play crucial role in different things. So what is the function of these DNA? So it helps in chromatin organization, gene regulation, evolutionary plasticity, and genomic arrangements. So now we will see that what are the different types of these repetitive sequences. To the classification, so repetitive DNA are broadly classified into two types: tandem repeats and interspersed repeats. So tandem repeats are where repeats are placed one after another, and interspersed repeats where repeats are scattered throughout the genome. Tandem repeats are further, their examples are satellite DNA, which is present in centromeres, mini satellite that is found in the telomeres, and microsatellites, which is very short repeats used in DNA fingerprinting. So like sat DNA, mini sat, and microsat are examples of tandem repeats. Interspersed repeats are scattered randomly. Think of them like popcorn spread all over your room, and they include signs, lines, retrotransposons, and DNA transposons. So signs are short interspersed nuclear elements, which is non-autonomous, and lines are long interspersed nuclear elements, which is autonomous, means they move on their own. And then retrotransposons move using RNA intermediate, and DNA transposons move using DNA, that is the cut and paste mechanism. Heard about junk DNA? So earlier these repetitive DNA were called junk DNA, but these sequences now serve as key indicators of genomic history, structure, and adaptability. I will post the quiz for this session soon, so just stay tuned, subscribe, and then solve the quiz also. And after that, in the next video, I'm going to talk about all the important questions on transposons and these repetitive sequences, and a mind map on jumping genes and transposons. That what is important from CSIR point of view. What and all I have taught you till now is very important. You have to remember that always, but along with that you have to study the mechanism of these interspersed repeats in detail, where the enzymes are present, which is autonomous, what are the examples of that, and